Well, hello and happy Tuesday. I hope you enjoyed working with the designer genes activity, and I hope that a lot of that started to get some ideas in your head or bring out some questions then that you have about genetics and how it works, because that's where we're headed over the next several days. If you are not 50% your dad, 50% your mom, and genetics doesn't work that way, which it doesn't, then how does genetics work? And how is it that we can be relatively confident in our predictions about how the next generations are going to come out? Well, right here you have this, and we're going to go over and organize our thoughts on how genetics works. What did we see in the designer genes activity that clued us in onto how this stuff works? Okay, so first of all, let's kind of dive in here. Okay, the word inherit, okay, when we talk about inheriting from the previous generation, we're going to use this word to mean that traits being passed down from parents to children, parents to offspring. Okay? Now, under this idea of traits, we need to remember a few things. First of all living things are made of cells. Okay, that's easy. Each part of our bodies has different kind of cells. We really started all of our science this year with the idea of how these cells that we have in our bodies, and all animals and plants have these cells, how they reproduce. They, in general, use either mitosis or meiosis, or depending on the cell in that organism, an organism can use both. Okay? It just all kind of depends on what cell we're talking about and what organism we're talking about. Body cells, remember this is the pizza story here, from one you get two, okay? They are diploid. They have matching pairs of chromosomes in those cells. So for humans, right, they have 23 pairs, or if you count them individually, 46 chromosomes in the nucleus. So, okay, so we pulled, well, I'll get there in just a second. Okay, so this body cells, skin cells, muscles, nerves, blood, bone, all of those types of cells are the body cells. And they reproduce using mitosis, right? From one that has 23 pairs of chromosomes, the chromosome numbers double, and then it splits, and now you've got 23 pairs here and 23 pairs in this new cell. So they are diploid. They have matching pairs, complementary pairs of chromosomes, the right numbers, to bring about this new daughter cell or this next generation cell. Okay? But the other cells, the specialized cells in the body of many organisms, but not all, sex cells, they're haploid. They have only 23 single chromosomes in the nucleus. Just like we were talking about before, when we were talking about opening up your closet or opening up the downstairs closet by the door and finding shoes, right? If a body cell is opening up that closet and seeing 23 matching pairs of shoes, I mean, every pair has a left and a right shoe. You got 23 pairs of those shoes. Sex cells are opening up that door and finding 23 left shoes. Maybe fashionable, but not very functional. Okay? In females, the cell that has just 23 left shoes, right? Those are called the eggs. And in the male of the species, they might have 23 right shoes. Okay? And the sex cell in the male of the species, those are the sperm cells. When you have these 23 left shoes combine with these 23 right shoes, then you get a beginnings. We call it a zygote. In many veterinary or medical terms, you might call this a fertilized egg. Okay, and this fertilized egg has now 23 complementary and matching pairs of those chromosomes. Okay. So that's a review of mitosis and meiosis. There's meiosis. here's mitosis. Okay? It really does. Genetics just falls right after our conversations about mitosis, meiosis, and asexual and sexual reproduction. Where's all this stuff found? Well, we talked about that already. It's in the nucleus. Okay? The nucleus, we know where the nucleus is inside the cell. Chromosomes are long strands of genetic material in the nucleus. You saw this when we did our DNA extraction. You saw this. Let me see if I can bring up some examples here. I should be able to. And here we are. We have them. So, remember these? Here are the pictures. There's DNA. Right? There it is. Let me bring this up big so you can see it. There are the pictures. You found the DNA, those long strands of genetic material inside every living cell, right? Every cell of every living organism. There you go. There's the long, kind of clear, spaghetti-looking DNA, right? You were able to extract that the other day, okay? Now, where is it found? It's found in the cell. Chromosomes are made of this DNA. So let me show you this in greater detail, okay? We've talked about chromosomes. Here is a matched pair of chromosomes, right? Here's one chromatid, here's another chromatid. Together we call those chromosomes. Well, you have 23 pairs of these. So let's just say that we're looking at chromosomes number 12, okay? And if we zoom in at some spot right here inside this matching pair, if you zoom in, that's where you're going to find this double helix-shaped spiral of DNA, deoxyribonucleic acid. We made a model of this out of those pipe cleaners just on Friday. Now, different sections, here's your DNA. Here's your DNA right here. That shape is familiar to us now. Now, when you made those rungs to the ladder of DNA, when you made the green or the orange, or the green and the yellow and the red or the blue, okay? When you made those, what you may not realize is you count up 2 or 3 of these rungs at a time. 3 of these rungs, each section of the DNA, those are called genes. And these genes are the genetic code that help to make you uniquely you. Maybe this section right here, maybe that section right there is a section that may be talking about or controlling hair color. One of many sections that control hair color. And we write down notes on what these genes are and how they're expressed. We call these little letters. These are the notes that we keep on what genes are present in the organism. If these genes are going to activate something that shows up in the individual, we write a capital letter. And if these genes are only expressed if there are a complementary set of lowercase letters, then we write these here. And both of these letters are called alleles. So let's go back to our paper and try to make sense of this now. So here we are. The chromosomes are made, right? We did that little zoom in of the chromosome to see that double helix shape. Chromosomes are made of DNA, deoxyribonucleic acid. This is the model that you made on Friday. Okay? Now, there we go. Chromosome theory of inheritance. Chromosomes contain pieces of information, the 3 little rungs on the ladder. These are called genes. And these genes control physical characteristics of that individual. And you may find them across the pairs. And different letters mean different types of traits that these genes control. Hair color, eye color, skin tone. Whether or not when you clasp your hands like this, when you look at your thumbs, whether your right thumb is over your left or your left is over your right. That's genetically controlled. Yeah, go ahead, try it. Take your hands, do that. Now look at them. Which thumb is on top? For me, my right thumb is on top. That is a dominant trait. If your left hand is on top, that is what we call a recessive trait. But it's still genetically controlled. If you're not sure which one you are, make yourself do it both ways. One of them is going to feel disgusting to you. You're going to be like, oh, that's terrible. This is the way that I do it. Okay? A lot of times we just clasp our hands and don't even think about it. But that's genetically controlled. Also, widow's peak. Okay? I do not have one. I do not have a straight hairline here. As much of one as I still have. Okay? If your hairline makes a little triangle peak right there, if it comes to a little bit of a point, that's called a widow's peak. That is genetically controlled. That's a trait you have because one of your parents or both had it. Okay? So these genes tend to control a lot of information about you, about your physical appearance and the physical workings of your body. And you get them from the previous generation. Okay? Genes have different forms. You either have a form that's going to show up no matter how many times it's there, or it may come in a form where that trait will only show up if both of these are the same. And that's when I say both of these, I'm talking about the letters that we assign to parts of the genes that control things. And these letters are called alleles. A-L-L-E-L-E-S. Alleles. This is where you may have heard the words like dominant and recessive. Okay? There are two alleles, one on each of the chromosomes in the pair. So you know how, like we were saying earlier, this might be pair number 12? Well, you've got one area right here on this left chromatid and this area over here on the right chromatid. Both of these areas on both of these chromosomes will control the same things about the body. Maybe this section right here is about how long your eyelashes will be. I don't know. But it's something. Okay? They control something. The gene involved in the shape of the chin has two alleles. One that produces a smooth chin and one that produces a cleft chin. Isn't that neat? Okay. Now, here's where you might be wanting to know about the words that you heard people use during our Designer Genes lab, and maybe you've never heard these words before, but now we're getting into the definitions of these. There's a word that means when the alleles of the gene that will be shown or expressed whenever that allele is present. And it's represented by a capital letter. So when a gene is represented by a capital letter, we call that dominant. What does dominant mean? It doesn't mean it's the strongest. It doesn't mean it's the one that's going to show up most often. That's a common misconception. Well, the dominant one is the one that's always going to show up. No. In Sweden, 96%, 94% of the population is blonde hair and blue-eyed. Blonde hair and blue eyes are both recessive traits. Yet, a very, very high majority of the percentage of the people in Sweden have blonde hair naturally and natural blue eyes. Those are recessive traits. So dominance has nothing to do with the number of times you see it out in public, like you see it expressed on an individual. Like, brown eyes are dominant. Well, they may be, but not because a bunch of people, not because the majority of people are showing that trait. Dominance and recessiveness have nothing to do with the population percentage that are showing that trait. Here's what dominant means. Dominant means that that trait, okay, is going to show up if both letters are, if either letter is capital. Okay? Like up here we talked about in this picture, right? Two alleles control a gene. Okay? Here's one, a capital and a lowercase. That's how we write it, right? You could write it like this, RR. Here's one, SS. Here's one over here, little t, big T. A dominant trait is one that if either one of these is capital, that's the trait that's going to show up. That's what dominant means. It could be the first allele, it could be the second allele. But as long as one of those are capital, the trait assigned to that capital letter is going to show up. Let's say in this case, this capital letter means green, and a lowercase letter means brown. Okay? If these are the letters that show up, one of them, it doesn't matter which, if one of them is dominant, the trait that's assigned to that letter is going to show up. So what color is this? Let's call it a bug. Is this bug going to be green or brown? Well, here are the genes that control the color. One of them is capital. We call it dominant because as long as one of them is capital, that characteristic is going to show up. So if this letter means green and that letter means brown, if I see a capital letter there, this means that bug is going to be green, no matter what this second letter is. The idea that as long as one of the letters is capital and that's the trait that's going to show up, that idea is what defines the dominant trait. Dominant. The alleles of the gene that will be shown or expressed whenever that allele is present. It could be just one dominant on the left, or the right one could be the capital. Doesn't matter. As long as there's a capital in there and that trait shows up, that's what we call dominant. Blank, the allele that will not show when there is a dominant allele present. Right? So let's look back up here. If this is dominant, this allele has a trait connected to it. This trait says brown. But see, the dominant one is the one that shows up. What do we call this one that doesn't show up if there's a dominant present? We call that recessive. We call that recessive. And furthermore, the recessives are only going to show if both letters are recessive. In other words, if you wanted that bug to be brown, you were just hoping for a brown bug, that combination of genes is not going to give you a brown bug. That combination of genes is not going to give you a brown bug. That's going to be green because I see a dominant color. I see a capital letter there. If both of them are capital, that's still going to give you a green bug. The only thing that's going to give you a brown bug is if both letters are recessive. Both letters are lowercase. That's how you get a brown bug. Okay? So recessive traits are only going to show up if both letters are lowercase. Even if you have just one capital letter, doesn't matter if it's that one or it's that one, that trait associated with the capital letter is going to be the one to show up. And when I say show up, I mean that's the physical trait you're going to see on that person or that animal or that plant. Right? So if you were this controls green, that controls brown, but this one's capital letter. The bug's going to be green. Sorry, brown, your trait just doesn't get expressed. Your gene is still there, but this is the one that's shown outwardly, and this one doesn't get expressed. This one is the one that gets shown outwardly. This is not the one that gets expressed. Both green. Because there's no capital here, then the brown will show up. You will see a bug that is brown thanks to this code, this genetic code. Okay, so there are two types of traits that we're going to talk about. The expressed trait, which is the physical characteristic that we see on the individual. I can see brown hair. I can see freckles. I can see on you the cleft chin. Okay? What do we call that? the physical trait that I can see on a person, the physical, pH, the physical, that we have called, that's the phenotype of a person or an animal or plant. pH, that's how I remember what phenotype means. pH, phenotype, the physical characteristic. I can see blue eyes. I can see widow's peak. I can see right thumb over left thumb. I can see those traits, right? I can see coloring of skin. I can see color of hair. Okay? Those things. Those are the physical characteristics on someone or something. We call those characteristics a phenotype. So if I asked you, if you saw a bug with this gene combination, what would that bug's phenotype for coloring be? Your answer would be green. Because a green bug, I can see the color of that bug. That's the bug's phenotype. This gene combination would give me a green bug. What is this bug's phenotype? Green. What about this bug's phenotype? Brown. Brown. Now, can I see GG? Can I see the genes inside that? No, I can't. No. But what I can figure out is this next word here on number 4. If I have letters written down because of a blood test or some other examination of the animal or the person, and if I can actually literally go in and look at the actual genes themselves, if I can go in and look at the genes and find the code, now you're not going to zoom in on DNA under a microscope and find, Times New Roman RR. You're not going to find it. That's just science has decided to put letters that stand for little stripes of DNA. Okay? But what do we call those little letters? If I gave you a question like, if you see a green bug, what would its genetic letters have to be? Or if you see a brown bug, what letters or what genes would that bug have to have in order for it to be brown? That's a way to think about it. Well, if I asked you that question, your answer would be, well, I know that the brown is 2 lowercase g's. What do we call that 2 lowercase g's? We call that a genotype. A genotype. Genes. Genotype. The root word there being genes. G-E-N-E-S. I can't see a person's alleles. I can't see a person's DNA. But if they're walking around with blue eyes, I know that their phenotype, their genetic code, has to be recessive letter, or that's their genotype for that trait. Okay? And you actually saw these things in the mice and in the chickens. Do you remember seeing these letters next to the mice? You saw the genotype of letters, and then you saw their phenotype, the coloring of fur. This genotype expresses black fur. How do I know that? Because pure black had these letters. So anytime a capital letter would show up for that gene, that tells me that that mouse is going to have black fur. This is that mouse's genotype. There are the genes. The coloring, the actual color, black fur, that is this mouse's phenotype. Black fur. 2 capital F's. White fur is the phenotype. What would the genotype be for white fur? 2 lowercase f's. Okay, that's how we write it down. We write the genetic code that would translate to the physical features we see. Now, it's going to take a lot of time if we had to, every time we talked about what are the genes of this organism, it would be a whole lot longer for us to say capital letter, or lowercase letter, or one capital and one lowercase. So what science did is it found a way to shorten that and use some science jargon to mean these different combinations of letters. Okay? Now, this word, heterozygous means that their genetic code is going to show one of each, one capital letter and one lowercase letter. So from this point forward, we're going to use this word heterozygous. And what does that mean? It is a way to talk about a genotype where one is capital and one is lowercase. So from this point forward, when I ask you questions about what are the possibilities of getting a heterozygous mouse, okay, you're not going to be able to say, well, that's one capital and one lowercase. No. That is now heterozygous. So when you see or think or hear the word heterozygous, what does that mean? The genotype that we're talking about is one that has a capital and a lowercase. Now, what if both letters in the genetic makeup are the same? Either they're both capital or they're both lowercase. We call that homozygous. Homozygous, when both letters are the same. Okay? So you're not allowed anymore from this point forward, if I ask you a question, you're not allowed to answer capital F, capital F. I will say, no. What do we call that? And you are to reply, oh, that's homozygous. Okay? Now, if you say homozygous, right, there's two options here. We have homozygous could mean they're both capital. Homozygous could also mean they're both lowercase. So how do we distinguish between the two? Right here. Right here. It is not enough to say homozygous. You have to tell me which letter will there be two of. Homozygous dominant means that the genes are both uppercase, right? The genotype is both uppercase. Homozygous recessive means that you're talking about a genotype that has two lowercase letters. So this is going to take some getting used to because these are new words that we're using, and that's okay. We're okay with that because we're going to have a lot of practice. You are learning a new skill. Okay? Skills take time. Let's do some practice right now. What I recommend is to keep this page out right here that says the homozygous, dominant, homozygous, recessive, and heterozygous. Just keep this part handy so whenever I say what I'm about to say, you know what to write down. I would keep that handy while we're going to work with some practice here. And what is this practice about? Well, we call these things Punnett squares. And here's why we use them. Just like in the mouse genetics gizmo, you took a pure black and crossed with another pure black or a pure white, and then you started hitting breed. Okay? Well, the letters next to the mice, we could have used that to accurately predict the chances of the offspring coming out a certain way. Okay? Here's how we do that. We use this little device called a Punnett Square, and it's very simple to do. Let's say we're going to cross a black mouse with another black mouse. But both of these black mice are heterozygous. Okay? Now, here's what we do. You know that that is heterozygous. And in the mouse gizmo, you know that these letters still gave you a black mouse. So this is one mouse. Here's another mouse with the same alleles for heterozygous. Okay, what are the chances that these two mice are going to produce black mice or white mice? Well, here's what we're going to do. We're going to take the two mice genotypes and we're going to plug them in to a little device called a Punnett Square. So here's what you do. You put one parent's genotype on the top. It does not matter which parent you put on the top. The other parent's genotype, you're going to put down the left-hand side. Okay? So let's do that together. You see where I came up with that? I'm using the genotypes of both parents. This parent, I just picked, it went up here. One letter over this column, the other letter, the second letter over this column. And that's fine. And that's fine. I chose the other parent, and I put the first letter outside of this row, and then I put the second letter outside of this row. Okay? So this is parent genotype number 1. This is parent genotype number 2. Now, from here, I have set up properly my Punnett Square. Now, we are going to, what I like to call, run the cross. Here's how you run a cross. This letter is the captain of this column, which means it gets written in every box beneath it. So let's write that down. This letter gets written in this box, and it gets written in this box. This letter gets written in every box below it. This letter is the captain of this column, and it also gets written in every box below it. Okay? This letter gets written in every box below it. It gets written here, and it gets written here. Good. Good start. Now, we have to then work with the genotype of the second parent. This letter is the captain of this row. And this letter gets written in every box to the right. So this letter gets written here, and this letter gets written here. It does not matter which side, right? It's not like, oh, all the dominants have to be, all the capital letters have to be on the left. It doesn't matter. That means the same thing if these two letters were switched. It means the same thing. This letter is the captain of this row. Okay? And you are going to write this letter in every box to the right. So this is going to get written here. See? And this combination means the same as this combination. And this letter is going to get written here. Science! Just making sure you're awake. Okay. So here are all the possibilities that one offspring could come out with. Okay? There are these possibilities. There are two ways that we write down these possibilities, because we're trying to use this little tool to help us predict if these two parents crossed, what would the outcome look like for the offspring? Okay? There's two ways that we write this down. First, we write down the genotype results. Then we write down the phenotype results. Okay. Let's look first at the genotypes. Let's start by looking at this first pane of the window. I see homozygous dominant as a result. How many other homozygous dominants do you see? I see no other ones, okay? So, I see 1 out of these 4 being homozygous dominant. What is 1 out of 4 as a percent? 25%. So there is a 25% chance that this offspring, as a result of a cross between these 2 mice, there's a 25% chance that that offspring will come out with the genotype homozygous dominant. Okay? But is that all that are in here? No. There are some other options. There's some other ways that this offspring could come out. In the next, so we started right here. Now let's look at the next window over, the next pane over of the window. I see heterozygous. I see heterozygous. And it doesn't matter which one's capital and which one's lowercase. This is heterozygous. Okay? I see 1 out of 4. How many other times do you see heterozygous? Oh, I see another one right here. So I see 2 out of 4, a 2 out of 4 chance that this offspring will come out with heterozygous genotype. Well, what percent is a 2 out of 4? That's half. Okay? So there's a 25% chance this offspring is going to come out with this genotype. There's a 50% chance that the offspring is going to come out with heterozygous genes. I see 25, I see 50. So I had both these. I see 70. So I've got 75% of the 100% chances I should find up here. So we've talked about this one. We've talked about this one. Here, I see homozygous recessive. This is a possibility. Homozygous recessive. Do I see any other homozygous recessives? No, I don't. So I see a 1 in 4 chance that this offspring could come out with homozygous recessive genes. What is the percent of 1 out of 4? 25. Okay? 25% homozygous recessive. Now, I see 25 plus 50 plus another 25. This equals 100%. I have now accounted for 100% of all the possibilities. So I am done writing down the genotype. If I add up these percents and they add up to 100, I have written down all the possibilities that could come from that cross. So I'm done writing down genotype. The offspring could come out, 25% chance of it coming out this way. That offspring has a 50% chance of coming out this way and a 25% chance of coming out this way. Before this offspring's even born, we have a good idea about the prediction for how it's going to look. Well, what its genes are going to be, I should say it that way. Now, when we talk about phenotype, this is where we're talking about what color fur would we actually see on the offspring. Let's look. Let's start in the same pane of the window. Up here, I see homozygous dominant. Now, in the mouse genetics, what color fur was attached to dominant letters? Black. Okay, now let's watch. Here, if this was the combination, this is black fur. Do I see any other combinations in this Punnett square that would give me black fur? So far, I see 1. Here's 1. There's 1 dominant letter in here. That means that black will show. So that's 2. No capital letter here. But there is a capital letter here. So that's 3. 1, 2, 3. What percentage is 3 out of 4? There is a 75% chance that this offspring is going to come out with black fur. So this leaves only one pane of my window left. And the phenotype, or the physical characteristics that are attached to lowercase letters, would be the white fur. So how many windows in here show a combination of genes that would give me white fur? Just one, this one. And so that means that this offspring does have a 25% chance of coming out with white fur. Now, the same rule when it comes to percent applies to phenotype as well. When I add up my percents and they equal 100, I am done writing down results. And there you go. Here are all the possible combinations. If a heterozygous mouse crossed with a heterozygous mouse, here are all the possible outcomes for that one offspring. It's got a 25% chance of coming out homozygous dominant, a 50% chance of coming out heterozygous, and about a 25% chance of coming out homozygous recessive. That same offspring also has a 75% chance of coming out with black fur, but it also has a 25% chance of coming out with white fur. This is how we use the Punnett Square in order to accurately predict the outcomes of crosses between plants and animals and humans. If you know the gene that you're looking for, and if you know what the parents have on that gene, say for example, you want to know if your child has the chance of having a genetic condition. Maybe you have the genetic condition and you want to know what the chances are of your child coming out with that same genetic condition. Well, what you would need to do is a little blood test, you and your spouse, you and your partner, I guess, and you would find what your genotype is for that genetic condition. Maybe it's a recessive condition, maybe it's a dominant condition, I don't know. But you would put your genotype here, right? And then the other parent's genotype right here, and you would run this cross. You would be a geneticist. And then you would come out with the information that you would need. A lot of times people will go to a medical professional and ask for blood work so they can determine, okay, what should we prepare for? You know? So that's how they use this very tool. Okay, in order to find out, or at least what the probabilities are of things coming out a certain way. Okay, you're about to run through more practices, and you're going to use these other Punnett squares to do those practices. You're going to set up the crosses. You're going to then run them yourself. You're going to come up with the genotype and phenotype results on your own, and then we're going to share out to see if everyone got them. Okay? All right. Good luck.